Okay, I take that as the key.
Yes.
Hello, welcome everybody to the second session of Transport Dementia 4, where now I would like to start the session about methods by speaking a little bit about how we develop custom deep learning algorithms to analyze histological images. And for that, we use the software DeePathology STUDIO. Yes. Histology is quite an old method. It has been around for a while, but it is still very relevant today because it is quite simple to do. There is a wide variety of stainings available. And most importantly, it includes spatial information about proteins, cells, and whatever. However, the quantification of histology can sometimes be a little bit challenging. So how do we quantify histology if you want to get from a nice histological image to a graph with bars and numbers? Of course, the first thing that people did was quantify manually. This is very time consuming. Imagine you're having to count all the brown dots on this slide. It is also less reliable, especially if you have more complex tasks where you don't only want the number, and you have a very strong researcher bias because it will depend on who counts the brown dots in this example, what result you'll get. So therefore, in our lab, we have been using for many years already semi-automatic analysis. This is in our case done with Axel Vision, but it can usually be done also with other imaging software. It is based on staining intensity, but here you already have the first problem. If you have variations between runs of stainings, that can be problematic. And usually for that, to correct for it, you involve some manual steps. And that again makes it dependent on the researcher. So now we have a new method. It is deep learning assisted quantification, which is a very fast method, especially with very little hands-on time, especially once your model is already established. And what is also nice is that even though it may make some mistakes, they're always systematic and reproducible, no matter who analyzes the data. So if you have a mistake, it will affect all slides from all groups, from anything. So that in that way, we get rid of quite of a big part of the researcher bias. Now, the issue is that, of course, we're not all computer scientists. And for that, DeePathology STUDIO offers an interface that where you can develop models without knowing any of the computer science behind, and you don't ever have to touch code if that's what you're worried about. So what does deep learning mean? Deep learning is a type of machine learning, and it is a subfield of artificial intelligence. And basically it means that a machine extracts knowledge from given data to make decisions for which it is not explicitly programmed. So how you usually use a machine is you tell it if A happens, then do B. But then when you come and you have situation C, it doesn't know what to do. And deep learning overcomes exactly that problem. So in our case, we're using supervised machine learning. And that means that it needs a certain input from us as a user. In general, you will find two phases. The first one is the training phase, where you show the machine images, for example, of strawberries when they're in season. And from that, the machine will derive a model to how to recognize strawberries. And this model you can then use in the prediction phase, where you will show a completely new image to the machine. And using the model that was developed before, it can extract objects called strawberries and also everything else, so the background. So a little bit more about the specific software that we're using. As I said, it's called DeePathology STUDIO, and it has a wide variety of modes. You have the regions mode, where you can basically separate and segment a slide into different regions that have a different cell concentration or a different structure in general. You can use the cells mode to quickly identify different types of cells. You can use the objects mode, which is the most versatile one, and the one we're going to focus on today, where you can basically detect any object, anything that you can see on a histological slide. And there's also the tiles mode, in which your slide is basically separated into smaller or bigger squares, and each one of the squares will then be classified as into a certain category. So in the objects mode, you have many different possibilities. I will just show a few screenshots from algorithms or models that we use in our lab. So the first and most used one is to detect plaques, where we have two categories detecting dense and diffuse objects, plaques. You can also use it to detect microglia. For example, in this case, it is trained to only recognize the cell body of the microglia. You can also detect astrocytes, where you can make it, in this case, it was trained to outline the entire astrocyte. Or you can use even just hematoxylineosine stains, and for example, train it to recognize nuclei with these pink inclusions. So now I hope you're all curious and you want to develop your own model using this software. So what would be the first question that you want to ask yourself? It is, what do you actually want to detect? And I'm emphasizing that because it's basically the most important question. And it is very important that you give clear input. As I said before, it is supervised learning. So the machine depends on your input to learn properly. It's like children. If you teach them wrong, they will do it wrong. And of course, this can be very easy. For example, if you have different colored berries on a green background, it will be very easy for you to make the correct decisions. But if you think about maybe all red berries on a slightly different red background, you can already see that this is more difficult for you as a human, and that will make it also more difficult to teach the machine. Next, I would like to show you a little movie that I've recorded beforehand, where I basically set up a sample teaching to detect amyloid plaques on this brain section. And I will play the movie. Hopefully it will work now. Yes. So first, if you want to create a project, you select the objects mode, you have a new folder, and you add a slide to your project. I did that really fast. You define your category or multiple categories. In my case, I just chose one called plaques. And you can go through the settings now. You can also do that later if you feel that you need to adjust certain settings. And then you're ready to start. And first of all, you need to make annotations to teach the machine what is it that you want to detect. And in my case, as I said, it will be plaques. So I'm outlining a couple of plaques or objects on the slides that I define as plaques. And you will see in the end, I will also annotate some background, and that is basically everything that the machine is not supposed to detect. So it's not common background, what you would think in histology, where you have a background staining somehow, but it's basically everything you don't want to detect. And once you have enough objects added, the machine will start training. And then what you can do is you can watch the progress. And in this graph, you see lower numbers are better. And very quickly after we started, the model was very much improved. So if you're happy with it, you can actually test it. So you just go on to a part of your slide and you let it run, and it will outline in yellow all the plaques that it has found. And what I then do is I add these plaques also to the training data set. So I save the time, but I don't have to outline them all manually again. I can just click on them, and then they will also be used to improve the model. And if I now go back to monitor the progress, you see that, oh, first the model got worse, but this is because I gave it more information that it first has to implement before it then actually gets better again. So now let's say you're happy, you have gone through a few rounds, added more objects, waited a little bit until it was adapted. Then what you can do is you can actually freeze the current status. So it's very simple, you freeze your model and nothing will change anymore from this point. So then you can use it to analyze slides. In this case, I just mark a region of interest. You let it run, it will detect here in yellow all the objects, and it will then generate an Excel file which contains summarized statistics, also some screenshots to show what it actually detected, and the raw data for each and every object that it has detected, which you can then use to analyze it further, for example, using Excel. So there are also a few other tools that you can use in the software that I didn't show in the movie, but I would briefly like to mention them now. And the first one is annotation review. So of course, if you give a lot of input, and that's basically what you're supposed to do, give it as much input as possible, then obviously mistakes can happen. And the annotation review are a very good method to look at your entire training data set and identify potentially mislabeled objects, or also just to get an impression what you actually gave as good examples. The next one is version control. So sometimes it's good to have a little backup before you continue. So then you can just make a snapshot that you can later on then import again and maybe continue at an earlier time point if your model went, I don't know, slightly wrong somewhere. And last but not least, I would like to talk about validation, because obviously you want to somehow have a measure to say how well does my algorithm perform. And for that, you outline a region of interest, but this time a validation region. And within this region, it is your responsibility to annotate all objects correctly. So for example, here, I try to outline all plaques or all objects that I think are plaques. And then the machine or the software will compare their reality. So these are my validation annotations with its own predictions where the model sees objects. And based on that, it will calculate the specificity and the sensitivity of your algorithm. And just for fun, I ran this also over the little algorithm that I created in the video for demonstration purposes. And already after a relatively short amount of time, we had a specificity of 96% and sensitivity of 85%. So now that you have trained your algorithm using your own slides and categories, what can you do with it? As I said, you can freeze the algorithm. I think that's the first step to do, to preserve it. You can use it for batch analysis. So I showed you in the video only the analysis of a single slide, but you can also analyze 20 slides without ever having to return to the machine in between. And finally, you can also share the algorithm with your colleagues so that they also can analyze their slides, which we in our group are doing regularly, because if one has developed a good model, for example, the recognized microglia, then there's no reason why the other person should do the same thing again, if the slides are comparable. So I think that was it from my side. I hope I could give you a good little introduction into this method. And if you have any questions, I'm happy to answer them.
Are there any questions?
I think no.
I answered one question already in the question and answer category. There was a question about number of layers, network architecture, and activation function. So this is a tool for the analysis of histological slides. You can have different slide layers. That's possible. So any of the slide scanner slides that can be produced can be used and analyzed. You cannot measure networks in a way. So if you can label networks, you can label single cells.
No, but
Golgi state, for example, you can measure this, or activation status of something. It depends on what you want to measure.
I think what Stoyan meant is if we can influence which neuronal network is actually used, and we cannot. So this is predefined by the software, and the different modes that I showed you, they actually use different neural networks to perform their tasks. What we're doing. For example, the objects mode is using instance segmentation. And you cannot influence anything basically that happens behind the scene. Like, yeah.
Yeah, the ML machine in the background is not accessible for the user. So we can access it and change it, but it's not accessible for the ordinary user. Yeah. Martin?
Yeah, very interesting. I was wondering how well the model is working if you would change the situation of the slides a little bit. Like, for example, there was another question, the panelist, if you would use an immunofluorescence slide or maybe another microscope, does that change already so much that the algorithm has to be trained again? Or is there some redundancy that it would, for example, also detect microglia in an immunofluorescence slide?
No, you have to train it for your, I mean, the software just sees your images. So for example, in this case, it's trained, I mean, I don't know exactly, I can't talk to it, but I imagine it sees something brown and it's round and it has some contrast of the background. So it maybe goes for shape and texture and color. So obviously now if you would have an immunofluorescence slide, that would most likely be, for example, green on black or something like that, right? So that would obviously look very different and you would have to train it again. But the training is not something that takes incredibly long. As I said, and I showed you the sample stats for the demonstration training, this was something that I did, I don't know, in an hour, and I didn't sit an hour there and I was training it. Mostly it works also in the background. So you have to give it some examples, obviously, to start with, but then it can also just learn on its own and you could just come back once in a while, maybe you add some more, you see how is it performing, where does it struggle. You can use the validation also to see, is it rather having a problem with too many detections, so too many false positives, or does it have a problem that it actually misses many microglia? So you can focus your training on that a little bit. Yeah. So yeah, you have to do it for every image or every type of image. You have to redo the training.
Okay, there was a question about IF slides. Yes, of course, you can do any analysis of any slide with any staining, as long as your eye is able to distinguish the objects that you actually want to measure. So if you are not able to distinguish this, also the software is not able to do this. But as long as you can say, okay, this is a certain feature that I want to measure, the software can do this, either cells or smaller objects or inclusions or anything, or inclusions together with a nucleus, these kind of things, it's no problem. So if you are able to do this, or a pathologist is able to do this, then the software can also do this. Okay, thank you very much for the questions. And we will proceed to